Hi there, science fans. Welcome to another episode of Science and Other Stuff for Senior Citizens. My name is Dan Casciano. If you like these COVID-19 videos, please subscribe to my channel and ring the bell to be informed of future videos. In the last video, I described the recent advances to the Moderna and the Pfizer vaccines developed to eliminate COVID-19. In the present video, I will describe some of the recent advances to the Oxford-AstraZeneca vaccine and the Johnson & Johnson vaccine. This presentation includes the mechanism on how these vaccines work. This slide indicates the general description of how the Oxford-AstraZeneca vaccine works. Since the Johnson & Johnson vaccine is constructed in a similar way, this description applies to it as well. This vaccine is based on the genetic instructions for building the spike protein. The spike protein instructions are stored in double-stranded DNA and not in single-stranded messenger RNA molecule. The double-stranded DNA is part of an inactivated viral delivery system called an adenovirus. Panel A on the left describes the SARS-CoV-2 virus that contains an RNA genome that provides all of the instructions needed to infect the human cell. It is studded with the so-called spike proteins that the virus uses to penetrate a human host cell, in this case, a human respiratory cell. This diagram also indicates a small piece of the RNA instructional molecule that codes for the spike protein. This piece is colored brown. In diagram B on the right, just the instructions for the COVID-19 spike proteins are contained in an inactivated adenovirus. This adenovirus is a virus that usually causes the common cold. It has been inactivated by researchers and is no longer dangerous to the human. Researchers have utilized it as a vehicle to contain and carry the instructions for the COVID-19 spike proteins into the human cell. The instructions for the spike protein is contained in a double-stranded DNA. This DNA becomes the major component of the vaccine. This cartoon depicts how the inactive adenovirus enters the cell. The adenovirus is engulfed into a membrane bubble that migrates to the nucleus of the cell and injects its DNA into the cell's nucleus. Although the adenovirus cannot replicate in the nucleus, its DNA can act as a template to make messenger RNA or mRNA. This mRNA leaves the nucleus and then acts as a template to make spike protein. Panel A on the left shows the mRNA in the non-nuclear part or cytoplasm of the cell, and then uses the cell's protein synthesizing machinery to make lots of SARS-CoV-2 spike protein. The intact and fragmented spike protein migrates to the surface of the vaccinated cell, so the host's immune system, especially the cell's antigen-presenting cells, can engulf the protein, converting it to a specific antigen that is then displayed on the surface of the antigen-presenting cell. This is shown in panel B. This part of the immune system switches on the cell's alarm system. By raising the alarm, the vaccine causes the immune system to react more strongly to the spike protein. Part of this alarm system is the antigen-presenting cell that communicates with the helper T cells, warning them of the invasion by a foreign virus and educating that T cell to the properties of that specific foreign invader. The helper T cells activated by the antigen-presenting cell then signals the immune B lymphocytes to produce antibodies that will specifically inactivate the SARS-CoV-2 viruses. Many B cells are transformed to make many more B lymphocytes and many more specific antibodies to COVID-19. This is shown diagrammatically in panel A on the left. In panel B on the right, the antigen-presenting cells also communicates to another type of white blood cell called the killer T cell. These cells are trained to recognize the virus-infected cell and then kills it to prevent further COVID-19 virus production. The antibodies produced by the transformed B cells then recognizes the spike proteins on the surface of the SARS-CoV-2 virus. The antibodies latch onto the spike proteins, preventing the interaction of the virus with the host cell, preventing entry of the virus into the cell. This neutralization of the virus results in no further infections. This immune response has been presented more in depth in one of my earlier COVID videos. The Oxford-AstraZeneca vaccine requires 2 doses given 4 weeks apart. It is expected to obtain FDA emergency usage during the last week of December or the first week of January 2021. The Johnson & Johnson vaccine is being evaluated as a single-dose vaccine and is expected to complete its Phase 3 human clinical trial in January 2021. It is expected to obtain FDA approval for distribution shortly thereafter. Both of these vaccines induce robust immunity and memory T cells, suggesting protection from subsequent infection by SARS-CoV-2. Although vaccination is imminent, we must continue to clean or wash our hands often, avoid close contact with sick people, practice distancing 6 to 10 feet, avoid indoor and outdoor crowds, and wear a mask properly. We will continue following these CDC guidelines until we reach herd immunity. For more information about science, visit my website at www.dcasciano.com. If you have any questions or would like me to develop a video on a specific science subject, leave them in the comment section below or email me at dan@dcasciano.com. Be sure to like this video and subscribe to my channel and ring the bell so you can be informed when I upload a new video. Thank you for watching Science and Other Stuff for Senior Citizens. Bye.